Hello children, so today I am going to explain you about the concept of Mendel's monohybrid and dihybrid cross. Okay, so firstly you might be thinking what is this monohybrid and dihybrid. So monohybrid means whenever we are crossing two plants considering only one kind of trait. For example, whether the plant is tall or short, so that is a one kind of trait, right? If we are considering two traits, how are we considering two traits while crossing? If we are seeing two factors, whether the plant is tall or short, and the other thing which we are seeing is what is the color of flower, okay, whether it is white, purple, so there are two kind of traits, so that is dihybrid cross. Okay, whether the seed is round or wrinkled, if we are considering two kind of traits, then that is dihybrid, and if only one kind of trait is being considered, so that is monohybrid cross. Okay, so now just let me tell you some important meanings, okay, so that you can understand the concept better. So here this capital T, it is indicating the kind of gamete. Okay, so these are the gametes, capital T, small t. Okay, so now gametes, you know, the germ cells from which the haploid cells arise, which are gametes and which play a role in the formation of zygote in eventually an individual organism, right? So that are gametes. Okay, now considering the allele. So what is allele? So it is a pair of two contrasting characters. Okay, so here there is capital T and small t. So capital T, it is indicating that this gamete is responsible for the tallness of the plant, right? And this small t, this gamete is responsible for the shortness of the plant. Okay, so that is an allele, pair of two contrasting characters, capital T and small t, that is tall and short. Okay, so that is called as allele. Okay, so now let us move forward. Okay, so here let me tell you what is dominant and what is recessive. Okay, so dominant here is obviously the tall plant which will be there, right? That is dominant. Okay, so here capital T, it is a dominant allele and small t is a recessive allele. Why? Because this small t is responsible for what? This small t is responsible for the shortness of the plant. That's why it is a recessive allele. Okay, now this recessive allele, that is which is responsible for shortness of the plant. So when we can say that the plant is short, whenever both of these are homozygous. Okay, let me first tell you what is homozygous and what is heterozygous. Okay, any individual, it will have one pair of allele, right? About one trait we are considering here. Okay, so that allele, it can be capital T, or it can be small t. Okay, there is also a situation where we are having capital T and small t, right? So this is called as heterozygous condition. This condition is called as heterozygous condition. Okay, and these two, these are homozygous. Okay, the terms are easy, right? So whenever there is homozygous, whenever plant is homozygous with capital T, then obviously the plant will be short. And when it is homozygous for two small t, then also the then the plant will be short. Okay, now what about when we are having capital T and small t, then also the plant will be tall. What? Then also the plant will be tall. Okay, why? Because here also dominant allele is there, dominant gamete is there. Okay, in the absence of dominant gamete, only the plant is short. That is, for the plant to show any kind of recessive condition, it must be homozygous for the recessive allele. Okay, that is small t and small t. Okay, this is only the condition when the plant will be short. Okay, so now let me explain you. Okay, so in Mendel's monohybrid cross, what he did was, Mendel, he crossed a tall plant and a short plant. Okay, that is you can see the alleles, right? So what were the progeny? Let me tell you how we consider the alleles of progeny. Okay, so what is the probability? You know, the parent cells which are diploid, both of them contribute one gamete, okay, and they fuse to form a diploid cell, right? So this parent, capital T, it will obviously give a one capital T only, right? And this short plant, it will also give it. What is the probability? It can only give small t, right? So this, right? So only one probability is there that the progeny will be tall. Okay, there is only one probability, and the gametes are also having only one probability, that is capital T and small t, right? Now this capital T and small t, see, I have also shown the how it has happened. See, this capital T and small t, right? Sorry, both of them capital T. This I have shown here. This is Punnett square by which we can know what alleles the progeny will be having, right? So this capital T, this small t, this short plant will give this gametes, tall plant will give this gametes. Okay, so you can see capital T, small t. So I have written this here. So the cross is same for all the things, right? So all the progeny will be tall. Okay, this was the first cross, right? Now this progeny were self-fertilized. You can see now the self-fertilization is taking place, in which what will happen? These heterozygous gametes will be crossed, right? So you can see now the next Punnett square. Okay, children, so these are the heterozygous alleles. Okay, so now in the Punnett square, what you can see, this capital T, that will give the homozygous tall. Okay, this T, so that will give heterozygous tall, and homozygous short. Okay, so how I have made this? So you can see any parent, from both the parents, both of them will contribute to one gamete, right? So one gamete from the first parent and another gamete from the second parent. Okay, this first parent I have written here, second parent is here. Okay, both of them will contribute one gamete each, right? So and afterwards the diploid condition will occur. Okay, so like that we have crossed here, and we can see the tall to short ratio, right? So here we can see these three are tall, here these are heterozygous, capital T and small t. But which condition will be expressed if both of them are if the plant is heterozygous? Then obviously the dominant trait will be expressed. So that's why these three are tall, but here there is no dominant allele, only homozygous recessive alleles are there. So that's why the progeny will be short. Okay, so here I have written tall to short ratio. If there are 4 plants, then three of them will be tall and one will be short. Okay, so this is phenotypic ratio. What is phenotypic ratio? Phenotype means the outer external appearance, that is called phenotype. Okay, now comes the genotypic ratio of the same. So genotypic ratio means the plant with capital homozygous dominant condition, homozygous recessive condition, and heterozygous condition. Okay, that we will see. So capital T, both of them capital T, there is homozygous condition for capital T, that is only 1. Okay, now heterozygous are 2, and again homozygous recessive is 1. Okay, so that is being indicated. So this was all about Mendel's monohybrid cross. Okay, now comes Mendel's dihybrid cross. Okay, Mendel's dihybrid cross. Okay, before moving to this cross, let me just give you some information, that is In Mendel's dihybrid cross, Mendel considered two kinds of trait, right? Which were those two traits? Let me just tell you. Okay, so firstly, the trait which was there, that was the seed shape, whether the seed shape is round or wrinkled, and the other thing was the seed color. Okay, whether the seed is yellow or green. Okay, now so let me just give you this information. This is not in your syllabus, but let me just tell you. Okay, it will help you understand much better. So Mendel considered Mendel experimented onto the pea plant, right? So in this experiment, Mendel considered 7 kinds of trait. Okay, so which were they? First was seed shape. Okay, then seed color, flower position, plant height, flower position, plant height, flower color, pod shape, and pod color. Okay, so seed shape, if the seed shape is round, then that is dominant condition, and if the seed shape is wrinkled, then that is recessive condition. Okay, now seed color, yellow is dominant and green is recessive. Flower position, axial is dominant, terminal is recessive condition. Plant height, that is easy, tall is dominant, short is recessive. Coming to the flower color, violet dominant, white recessive. Pod shape, so inflated is dominant and constricted is recessive. Coming to the pod color, then green is dominant and yellow is recessive. Okay, so based on these 7 characteristics of pea, okay, Mendel studied the genetics. Okay, considering these 7 characteristics. Okay, here the doubtful condition is regarding seed color and pod color. Okay, so you must remember that if the seed color is yellow, then that is dominant trait, and if the pod color is green, then that is dominant. Okay, so here you need to be clear about this thing. Okay, so this comes in your 12th class syllabus, but I am just explaining you this to have a better understanding. Okay, now comes the Mendel dihybrid cross, which is given in your book. Okay, so Mendel's dihybrid cross. So in this thing, first let me tell you this. Here capital R, it means round when the seed shape is round. Okay, and small r, it means when it is wrinkled. Okay, now this capital Y stands for yellow color of seed, which is a dominant trait, and small y represents green color. Okay, so yellow color and round seed are the 2 dominant traits. Understand, students? Now, okay, understood. So now let us move. Okay, so firstly what Mendel did was he crossed a round green seed and wrinkled yellow seed. Okay, so here both of them are having one dominant and one recessive trait. Okay, here the seed is round but it is green. Here the seed is yellow but it is wrinkled. Okay, so now when Mendel crossed these 2 progeny, that is when Mendel crossed round green and wrinkled yellow progeny, the progeny obtained was this, that is round yellow. This is what? Round yellow. Okay, let me still make you clear. Okay, here see, here both of them, these are capital R. Okay, so the seed will be what? Round. Okay, and here this is homozygous recessive for green color. right? Because there is small y. Okay, so that's why the seed is green and it is round. Okay, and here it is homozygous recessive for wrinkled condition. Okay, so that's why the seed is wrinkled, and but it is homozygous dominant for yellow color, right? So that's why the seed is yellow. Okay, when we will cross both of these, okay, so here we are sure that the gamete which will come in progeny, that is capital R and small y. Okay, no other option because there is no heterozygous condition, right? So, and here also there is a homozygous condition, so that's why we are sure that this will contribute to small r and capital Y. Okay, so that's why the progeny which is obtained by crossing both of these, that is this, which is heterozygous. Okay, so this one is your F1 generation. Okay, and what kind of seed is this? Here there is capital R and here is capital Y, so means the progeny is having dominant condition for round shape as well as for yellow color. Okay, now this progeny is being self-fertilized. This progeny which is obtained, we will do self-fertilization. Self-fertilization means when the two same kind of progenies are being crossed, then that is called as self-fertilization. Okay, so when self-fertilization occurs, right, so that is between these heterozygous progenies. So what we will get, that we need to see. Okay, so again I have made a Punnett square for the same thing. Okay, so again we are having a Punnett square. Okay, so now let me tell you. So see children, this upside which I have written, that are the gametes being contributed by the parent. Okay, so here what are the probabilities that I have written? Okay, so this, talking about this one, obviously both the gametes will be same. Why? Because this is self-fertilization, both have the same genotypes. Okay, so let me just explain. So here what is the probability? Here from this two, any one gamete will come in progeny. Okay, so taking let us consider that this dominant R which is there, that is going. Okay, so this capital R, and from this also dominant one is going. Okay, so that is one condition. Okay, now this capital R and small y, these gametes can also come, so that I have written over here small r and capital Y, these gametes can also come, right? And this small r and small y, they can also come. Okay, so the same thing we will repeat for this thing which is the same genotype. That's why I have written over here the same kind of thing. Okay, so now let me explain you. Okay, so now we are going to learn about this Punnett square. Okay, so now here we are having haploid condition. Why? Because of these are gametes. Okay, these are gametes contributed by the parents. Okay, and this one which is there, these are also gametes. Okay, this is easy thing to identify gametes. Why? Because these are haploid. Okay, so these are gametes, these are gametes. Now we are going to see the probabilities. Okay, what kind of progeny can occur. Okay, so now crossing we will do with the help of Punnett square. Okay, so now if this capital R, capital Y, capital R, capital Y, okay, if this so we have crossed, so this thing will be obtained, right? So that is round yellow seed. Okay, this is homozygous for both the conditions which are dominant. Okay, so that's why this is round yellow. Okay, again crossing these two gametes. See children, now the crossing is easy. Just the thing is to put the gametes with their correct probability, right? So now crossing between these two. Okay, this is round and green. Okay, this is round and yellow. Okay, now crossing between this thing also we are getting round and yellow seed. Okay, so like that here crossing between these two we are getting round in yellow still we are getting. Okay, why? Because of this capital Y are there and capital R is there. Okay, so whenever we are having dominant allele, that kind of trait is only going to be expressed. Even here, because of crossing between this small r and y and capital R and Y, we are getting this thing. Okay, that is round yellow still we are getting. Okay, why? Because of in this case both of these are dominant which are being expressed. Okay, now taking the second one, that is this capital R and small y. When I am crossing this capital R and small y with this capital R and capital Y, also we are getting what? Round yellow condition. Okay, now in this case if we see, then capital R and small y, capital R and small y, right? So the seed is round but it is green. Why? Because there is no allele which is dominant for yellow color. Okay, so like that I have crossed all this. Okay, now you must be understanding better, right? So like that I have crossed the whole. Okay, if you want to refer from your book, then also let me tell you, just let me. Okay, so if we see from your book, then you can see the same thing which I explained, round and green seed and wrinkled in yellow seed, that is being taken as parents. Now when the gametes are being contributed, then we are getting the dominant phenotype, that is round and yellow colored progeny. Okay, now when these two are being crossed, okay, so we will take all the probable gametic pairs, right? So capital R and small y, from this capital R and small r, any one gamete will come from this parent. Okay, so if we are taking capital R and from this two also any one gamete will come, so capital Y. Okay, so from this is capital R and capital Y, like that also there is probability of capital R and small y. Okay, so all these probabilities here, these are written, same is being done over here. And now according to the Punnett square it is being crossed. The crossing is easy, right? Now then we need to identify, this is round yellow, this is also round yellow, round yellow, round yellow, right? The same occurs over here, round yellow, round yellow, round yellow, round yellow, right? Now here crossing between capital R and small y, capital R and small y. So here this one is homozygous recessive for green color, right? So that's why the seed is round green. This seed is round yellow, this seed is round green. Okay, now this one is also round yellow, this one is wrinkled but yellow. Okay, the same over here, wrinkled but yellow. This one is round yellow, this one is round green, this one is wrinkled yellow, this one is both the traits are recessive, that is wrinkled green. Okay, so that is okay. Now here you must be wondering what is written over here, 9331. Okay, so this is the ratio of the phenotypic ratio is being given. Okay, 9331. Okay, let me just explain you this thing if you are confused. Okay, see, so what is being given over here is the gametype. This is your genotypic ratio. Okay, if you see over here, these are, if we take this two, then 16 probable genes, 16 progenies are being obtained over here, right? If we see from the 16 progenies the phenotypic character, okay, their external appearance, okay, then round yellow, if we count for round yellow, then how many are there round yellow? 1 2 3 4 5 6 7 8 and 9. So 9 are round yellow, so that is written over here round yellow. Okay, now talking about round green. So how many progenies are round but green? So 1 2 and 3 progenies are round green. Okay, now talking about wrinkled yellow. So how many progenies are wrinkled but yellow? So 1 2 and 3 are wrinkled yellow, and wrinkled green is only one, which is both of the traits are recessive. So wrinkled green is only one. Okay, so this is your phenotypic ratio, the external appearance. And if we take a total of 16, then what probability can we see? That 9 will be round yellow, 3 will be round green, 3 will be wrinkled yellow, 1 will be wrinkled green if we take 16 progenies. Okay, but here 556 seeds are being taken. Okay, so according to that these numbers are being given. Okay, if you are confused about what are these numbers, then if we take 16 progenies, then these are the ratio, that 9 will be round yellow. But if 556 seeds are being taken, then this will be the ratio. Okay, this will be the numbers of the progenies with their respective characters. Understood, children? Now let me tell you. So again the same data, you need to learn this phenotypic ratio. Round yellow is 9, round green is 3, wrinkled yellow is 3, and wrinkled green is 1. Okay, now this one was phenotypic ratio. Genotypic ratio might not be asked, but let me just tell you for your information. Okay, so here talking about the genotypic ratio, first is capital R and capital Y when the progeny are homozygous for both round as well as yellow color. So how many are they? So if you see, for that only capital R and capital Y homozygous condition is being expressed only in one progeny. Okay, so here I have written 1. Okay, now homozygous for round color and heterozygous for homozygous for round shape and heterozygous for yellow color. So that is over here 1, and the other one is over here. Okay, so 1 and 2. Okay, so like that. Now homozygous for round color and heterozygous for yellow color, that is being also explained. That is over here, that is the first one, and the another which is there, that is over here. Okay, so like that this is genotypic ratio. Now you can make your own Punnett square and cross verify this information. Okay, so this is the genotypic ratio. Okay, so in your higher standards you might be asked such kind of questions. Okay, this is a bit of extra information. If you are not understanding, no need to worry about it. Okay, so a question might be asked in Mendel's dihybrid cross for 16 progeny, how many of them are homozygous dominant for round shape. So we need to find how many are homozygous dominant for round shape. Okay, so I have the ratio already taken. If you don't have ratio, you can also cross verify with your Punnett square. Okay, so if you see how many of them are homozygous for round shape. Okay, so this one is homozygous for round shape. Okay, means in short we need to see where there are two capital R. Okay, So this one is heterozygous, this one is heterozygous, this one is heterozygous, this one is dominant, homozygous recessive, homozygous recessive only. So there are 3 which are homozygous dominant for round shape. These are 3 genotypes, okay? But how many are they? See, this genotype is with 1 plant, this genotype is with 2 plants, and this genotype is with 1 plant, okay? So in total 2, 3 and 4, okay? So 4 out of 16 are homozygous dominant for round shape. If you need to calculate the how much percentage are homozygous dominant for round shapes, then that is also easy, you must be knowing, right? So this is the case if they are asking us the percentage. So 25% of the plants are homozygous dominant for round shape, okay? This is a bit of a tricky question as compared to your level, okay? So no need to study it so well, okay? You will be learning this in your higher classes, okay? So that was all about Mendel's dihybrid cross, okay children? Now in your book there is also another thing given, that is about so let me just explain you that concept. Here it is given about this flowers, okay? There is no information given in your theory part, but this is only the picture is concept being given, okay? This also you will study in your higher class, but here only picture wise concept is being explained, okay? So here what they are taking is violet flower and white flower, okay? So here as I told you earlier, violet is dominant trait and white color is recessive trait, okay? So if I take this as homozygous dominant and this as homozygous recessive, then I will get progeny which will be something like this, right? So obviously it is also having violet color, okay? Now self-fertilization of this thing, when it will occur, then we will get 3:1, which is same as the when we took about tall and short plants, okay? So I have explained the same concept over here. Here capital W indicates purple color and small w it indicates white color, okay? So when these two are being taken, then obviously this is only the probable genotypic this is only the probable genotype which the progeny will be having. When this thing is being self-fertilized, then I have made again Punnett square. You can see here 3 will be with white colors and 3 will be with purple colors and 1 which is there, that is white, okay? So phenotypic ratio is your 3:1 and genotype is also same as you did earlier for tall and short plants, right? That was 1:2:1. 1 is for homozygous dominant, 2 is for heterozygous, and again 1 is for homozygous recessive, okay? So that was all about the crosses which are given in your textbook, okay, about Mendel's dihybrid and monohybrid cross. That's all children. For more videos please subscribe to my channel to get notifications about each and every video. So thank you everyone and bye bye.